Okay. Okay, guys. Can you see me? Can you hear me? You can hear me?
Yes,
All right. Thanks, Kelsey. Hi. How are you, little buddy?
I'm good. How are you?
I'm doing well. Let's do this thing. So we're going to talk macromolecules. All right. Um, with a little bit of pace. Wow. I don't want that slide. I want to go all the way back. All the way back. Macromolecules, lit chemistry, and life chemistry. Okay. So we're going to focus on polymer structure, monomer structure, elements in each, and then their functions too. Okay. So first off, life is based on carbon. Deal? We originally, erroneously, I still have my organic chemistry books from college. They were happy times. I loved organic chemistry. Uh, so I did send them back. So if you need an OChem textbook next year, let me know. But anyways, um, they thought that life chemistry was different than chemistry of materials, than rocks. They're wrong. Okay, it's all the same rules, but we've stuck with these arbitrary divisions of OChem, organic chemistry, and general chemistry, right? Well, today, for you to be a part of an organic molecule, you've got to have essentially two elements as your backbone. Number one is carbon. Okay, carbon is the backbone of organic molecules. All right. Organic molecules are generally made of carbon and hydrogen backbones. For you to be an organic, those are the two things we're looking for. All right. Now, there's other elements that are also, going to make up organics, right? I think you learned CHNOPS as an honors bio kid, right? Carbon, hydrogen, nitrogen, oxygen, phosphorus, sulfur. But here's the deal. Anyone know, if you want to unmute yourself, if you can, or throw it in the chat, what kind of molecule is this? Like, which macromolecule is this one that I'm circling right now? If you know it, say it. If you want to type it in, go for it. We got a chat here. Let's hear. We got glucose. All right, Hannah. You're right. And whether or not it's glucose, well, we don't get to see the double bonds, but it's a sugar, right? What makes a sugar a sugar? What makes, this is a hydrocarbon, which eventually, we make methane. Okay. How about this one? Anyone know what this one is? Throw it out there in the chat, or you can unmute yourself. 3, 2. All right, we got a chat. We got an answer, and it's amino acid. Attaboy, Ethan. Welcome to the party, buddy. You're right, amino acid. You notice the nitrogen is in the amine group, and here's your acetic group, your acid, okay? We got more chats. Let's see here. Yeah, Ethan, says Kurt. Attaboy. Okay, this is fun. All right, in a way. Okay, so amino acid. Okay, here, last question. What do we call this part of an amino acid? What is this deal right here?
The R group.
All right, good. Beating the typers. Yeah, that's the R group. So this is like your variable. We'll get to it in a minute, but this is like your X and your in as far as variables go. All right, macromolecules. Polymer, monomer. Okay. Who knows what's the name of the type of reaction when we take monomers and combine them to make polymers? This may have been a while. This is more like trivia than necessarily a question they'd ask you on the AP exam this year, but they might use the term, so I want to just throw it out there. 3, 2, 1, 0. Okay, it's called a dehydration synthesis. I've got a picture of us using monomers to make a polymer. Okay, dehydration synthesis. Dehydration means taking water out, making polymers. Okay, so here's this handy little table. I got this from some textbook, but here it is. Amino acids, they make proteins. They're the monomer of that protein, right? Monosaccharides make up polysaccharides. Nucleotides make up nucleic acids. If you know your macromolecules inside and out, like, well, what about the lipids? Lipids don't quite have a true monomer. All right, and so I don't know if we call them a polymer, but they're macromolecules. They're bigger molecules. Okay, so just real quick, monosaccharides, CH2O. That's the general formula of a saccharide, right? Of a sugar, right? Carbohydrates, right? Carb, right? Think carbon. O is the connecting letter in biology and medicine. Hydrate, think water. Carbon is connected to two hydrogens and oxygen, CH2O. All right, so here's your monomer. Well, here could be starch. We find starch in plants. Okay, plants, if they're can store their sugars two ways, as cellulose or starch. Okay, well, the good news for us, when we go and eat potatoes, right, a starchy vegetable, there's a lot of sugars in there that are stored as starch. Okay, we have the enzymes, which we'll talk about depending on how timing goes. Okay, maybe we'll do it there, we'll call it a day, we'll see. All right, but we have the enzymes that can cut. Here's what digestion is. We just did it in that fizz. Digestion is taking a macromolecule and slice, 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 slice up these little guys into their monomers. That's digestion. Absorption is getting those things from my gut into my blood so my cells can use them. Okay, so digestion, we can digest these. You may have heard celery is negative calories. Well, the reason is we can use energy to chew that crunchy celery, not the soggy, nasty old celery. Okay, but anyways, and sorry for the sounds on the audio here. But anyways, once we can chemically digest these by separating them, we can't digest cellulose. I mean, while we can make it smaller with our teeth, we can't break it down into monomers. The monomers of cellulose, if we could get them, there would be so many calories in cellulose. But we can't break the bonds between the sugars and cellulose. Cows can, but it takes all freaking day and some specialized stomachs, right? So there you go. Here's just a quick list. I'll share this with you guys later. But plants use starch for energy, cellulose for structure. Okay, glycogen is our form of sugar storage, right? We store athlete alert. Okay, we store glycogen in our muscles. All right, we store glycogen in our liver, and that's about it. Glycogen is a sugar storage molecule for us, and it makes sense if you think about it. We store in our liver. Well, if our blood sugar drops, as long as we're not diabetic, if our blood sugar drops, our liver will slice up that glycogen, okay, and release it into our blood as glucose. So your blood sugar levels measure the glucose in your blood. Athletes try to manipulate their glycogen storage, okay? Well, how do we do that? So, I mean, if you guys are a part of any team, what do we do at Mayfield High School? Okay, we have pasta dinners, baby, right? We have big time pasta dinners, okay, on Thursday. Now, we are taking a little smidge of science and trying to apply it, and we're not doing it right, okay? So, but you know what? Most of America does that, okay? See your disinfectants, okay? But anyways, all right, okay, getting political, but whatever. All right, I can't see your guys' faces, so I don't know. But here's the deal. To carb load, you kind of have to starve yourself of carbs for usually a week, I think it is. All right, so you want to have generally no carbs for a week. Well, what that's going to do is, what we're hoping to do is get your muscle cells to maybe have emptied of their glycogen or maybe sucked up even more, and maybe I'd have to go. I got my, let's see here, strength and conditioning book right here, okay, as we strain for Nat Fizz and Pack. And I should just go and check it out real quick. But I'm not going to, okay? And I can't, but the process, what's that, say again?
Shameless plug.
Yes, thank you for saying it. Shameless plug. Take a Nat Fizz and Pack. This won't be your textbook, but this is the book I'm using to study and train and take my vitamins. And anyways, it's changed my exercise routine by getting ready for this class. But I digress. To carb load, you have to starve yourself of carbs. And then the goal is, once you're really hungry for those carbs, you're going to soak those carbs into your muscle and store them as glycogen, hopefully in your muscles for a nice long-term, Wednesday night game night, right? If you've got a Wednesday game, you have your meal about 24 hours beforehand. Okay, and that's why we do our pasta dinners, but we're doing it wrong because we're eating carbs all week. But anyways, let's move on. Okay, so proteins, they're made of amino acids, all right? I have no mouse. So here's amino acid 1, here's amino acid 2. You're going to get a question today on how do we classify amino acids, okay? And I almost want to get out of here, but I want to stay focused. Okay, but how do we classify chemicals? I'd ask you guys that. How do you think the chemistry department, they've got those storage rooms, right? How do we classify our chemicals? I'm going to pause and wait for an answer. How do you classify a chemical?
Reactability.
Okay, that's pretty good. Agreed. So all right, make that a little more now. Now tease that out. Reactive how, we might want to know. Reactive with what? What might tell us?
Oxygen.
Nope. So characteristics. So like how
With each other.
so what?
With each other.
Nope. It's okay. No pun intended. It might be more basic than that. Okay. Okay. But that's one of our classifications. How does Dylan Gamber classify people in his phone, right? Hotties, okay? Girls he might call later, right? And girls that have slapped him in the face, and dudes. That's how they're classified in Dylan's phone, right? He puts them in the company name, so he keeps track of who's who and what's what. Okay, but I got a message here. Let's see what do we got. Reaction to acid base. Okay, so that's it right there. One way we can classify chemicals are they acidic or are they basic? All right. Another way we're going to classify chemicals is are they polar or nonpolar? Are they ionic? Right, which kind of fits in well, acid and base, I guess, are ionic too. But that's how we're going to classify amino acids. Basically, are they polar or nonpolar? Are they acidic or basic? And sometimes are they ionic? Right. That's how we classify chemicals. And then Dylan's kind of right with, as far as us, if it's explosive, we've got it packed away somewhere safe too. So we've got the explosive deals, we've got acids, we've got bases, we've got salts. Okay, the ions. So there you go. That's how we classify amino acids. Now, these R chains interact with each other. Sometimes, these proteins are really long. They might a polar and a nonpolar amino side chain. These R groups, they're going to interact and repel each other. Okay, whereas an acidic side chain or an acidic R group and another acidic R group, they might want to hang out together. Okay, just depending on some deals. Okay, so where were we? Here's some proteins, right? Let's think about, and this is for what's the function of proteins, right? This just represents 3, right? Well, Rubisco is an enzyme. Okay, so sometimes I think the number one function of proteins is enzymes. All right, we taught you all enzymes are proteins, right? The function of hemoglobin is transport. So some proteins transport or movement, like your muscles are made of proteins, so movement. Communication, growth hormones are proteins. Okay, there's others, right? There's defense, your antibodies, they're made of proteins. Identification, antigens are often proteins. Structure, your hair, high in protein. Don't eat it, okay, but it's high in protein. All right. Nucleic acids we're on a third macromolecule. So they've got carbon as these little side chains, right? Hydrogen, oxygen, and phosphorus. Okay. Oh, wait. And I left out nitrogenous bases are in the nitrogen base, right? So the question is going to be can you describe to me, we'll ask you guys, and I'm not going to answer the question on the FRQ in case maybe you've already seen it. Describe the parts of a DNA base, of a monomer of DNA. Tell me about it. Go ahead, kids. I'll open up the chat too. Can you guys see the chat? I don't know.
Pentose sugar, that part of it?
That would be one part. One part
Phosphate.
of your DNA. Yep.
Nitrogenous base.
Great. So we got a phosphate group. We've got, you don't need me to type, do you? A nitrogen base. And then deoxyribose sugar is in DNA, right? You guys aren't reading that, right? I hope. I don't want to distract you. But ribonucleic acid is what's in RNA. We got more. Chat. All right, A, G, C, and T. Those are the bases in DNA. Good. I'm with you. Okay. Circle. There you go. Deoxyribose. Right. This is kind of nice. I kind of like the PowerPoints with no words on them and just pictures. Okay. You guys can go look stuff up later. All right. So here's just different pictures. Here's DNA. It's double-stranded. RNA. It's single-stranded. It has U in it. Okay. There you go. Just different pictures of it. Building the polymer. Again, it's a dehydration synthesis. Right. As we build these polymers to each other, okay, we're going to lose an oxygen, a hydrogen, and another hydrogen from these guys here. Say again? What are you talking about?
What?
I don't know. You said something. I don't know what it was. Okay.
My mother.
That's okay. Tell her we say hello. So there's your backbone of DNA. All right, last one is lipids, okay? So biochemistry of lipids, all right? What makes a lipid a lipid? Can I zoom in here? No, I can't zoom in. Darn, sorry. A lipid is a lipid because it's almost exclusively carbon and hydrogen, right? If I could just I can't. If I could just block this off, which you guys can't see as I do it with my hand. But notice here's a triglyceride. This is how animals store their lipids, right? This is an animal fat, right? If you have a burger or a steak or a pork chop or meatballs or bacon or lamb or chicken. Okay, I think you get the point. Okay, now I'm just having fun with you. So you would that's how your fat, the fat you get. This is typically how animals store their fat. Okay, triglycerides. This is how we store our fat. All right. So triglycerides got 3 parts. It's actually got a sugar backbone. This is a glycerol. You see these CH2O, right? CH. Well, there used to be an H here, right? But this used to be a glycerol sugar. Okay, so triglycerol. Sorry, triglycerides and triglycerol doesn't matter. Okay, they've got 3 fats, 3 fatty acids. There's an acid head to this fat. Here it is, right? This double O, and this used to be an acetic group, right? Well, get rid of that H, get rid of the O. What do we have when we combine monomers? Dehydration synthesis. Okay. Real quick, side note. What do we call? What's the process of breaking down a polymer into a monomer? Okay, I'm going to write it out so you can see the word. Get my chat up here. The process of, I don't know, can you see when I pull up the Zoom chat, can you see it? I don't know what you see. Okay, but the process of breaking down a polymer is called hydrolysis or hydrolysis. Okay, hydrolysis. This word basically breaks down to hydro, and remember, and I know the other part, but it's a connecting word. So water to cut. Using water to cut molecules, what we do is we're going to hydrolysis. If you imagine like adding a water here, right? So if we added an H here and an OH group here, that's how we separate this. That's hydrolysis. An enzyme could speed that up. Oh, is that it? Oh, can I not go for it? Oh yeah, good. I can say I have a lot more here on fats. I don't want to do much on fats. I listen. There used to be a time, guys, where sometimes I used to like to gamble a little bit. Okay, as a former gambler, no longer do I gamble, right? But I don't know why I'm telling you this. But as a one-time gambler who understood odds, I like our odds of not personally, right? I don't like the odds there being a biochemistry question on it personally, right? As far as polar and non-polar, I just think if you get 2 FRQs, polar and non-polar, they'd have you take AP chemistry, right? If they want to test you on that. That's my thought. You better, that's why I think the basic parts of these, we should be good to go with our biochemistry. I think all of this is fair game, but any deeper, now you're going to be in the chemistry world, and there might be a revolt or a revolution if they only ask you chemistry question on AP bio exam. Just my thought. Okay, so that's why we chose to do this first, hit it, and let's move on. Okay, saturated fats. All right, well, saturated fats, what are they saturated with? Hydrogens. Okay, saturated, be full of hydrogens. Right, look at these carbon side chains. There's not one empty space where that one hydrogen isn't there. So saturated fats are not good for you. Deal? This is about how they look when they stick to your arterial walls. You got atherosclerosis, right? Is that these saturated fats are super, super stable. This is why restaurants love to cook with butter. 2 things, right? Well, 2 things that that butter has going for it. As far as once your restaurants get back into business, okay? And as you're like, what did the Verdes buy? Now we bought some butter. He uses an olive oil guy, right? But why do we buy butter? It stays good for a long time. These are very stable. They can stack. They're not that reactive. Butter doesn't go bad for a long, long time. Okay? The other thing is they taste delicious. Why have we evolved to love the taste of butter? It has a lot of calories in it. Okay? There's a lot of calories. Let's see here. What is this? In 1 is that a tablespoon? Yeah, in a tablespoon of butter, we get 100 calories, right? Butter. Okay, a lot of calories in fat. Deal as far as per gram. Okay, so it tastes good and it stores well. That's pretty good. For when these restaurants come back into business, they're gonna use butter, baby. Oil doesn't stay good as long. I say oil, but unsaturated fatty acids. What I want you to notice is this guy right here. See this little kink right here? There's a spot for a hydrogen to go, but it doesn't have one. And same with this carbon. This is an unsaturated fatty acid. Here's the acid part. Here's the acid. Here's your fat. Fatty acids are the terms you hear all the time. Well, now you can see there's this acid head here. If only I had that hydrogen there that wasn't gone in the dehydration synthesis reaction, right? So what are they unsaturated with? Hydrogens. Okay, you can kind of think like this. Really, where's the energy that we get from our food? It's between the carbon and the hydrogen. Anytime we break those bonds, that's really where we get our energy from, right? You can think of like NAD and NADH, right? Separating a carbon from a hydrogen, lot of energy in there. Okay, well, fats have lots of carbons and lots of hydrogens. Unsaturated fatty acids, they don't. Why are they less good for you, right? Well, there's these low density lipoproteins and high density lipoproteins. They're these proteins that carry around fats in your blood, right? Low LDLs are healthy fat. They're low density lipoproteins. Basically, they can't carry that many fats. There's all these kinks, they kind of get in the way of each other. You can imagine if I'm going to stack 100 papers, I want those papers perfectly flat. Okay, if I'm going to stack like some, they're going to be crumpled and in the way. Okay, so if 100 papers are crumpled, they're going to take a lot of space and they'd be lower density. Okay, so anyways, saturated. Sorry for the formatting. Bad, good. Generally so. Everything in moderation. Okay, that's it. Well, we want to talk about my sister. All right, here you go. Phospholipids, and we're almost done, kids. Okay, phospholipids are famous. Our favorite lipid, maybe of all time. I don't know. Okay, but they've got the polar head. Loves water. Hydrophilic. Hydro meaning water. Philic meaning love. Philadelphia, the city of brotherly love. Right. Anytime you have philia, it means love. Hydrophobic, I think arachnophobia, right? My kids aren't scared of spiders at all. Good for them. They know they're eating the ants. Okay, let them live. All right. But anyways, hydrophobic, they're afraid of water. So fats hate water. All right. We've talked about the cell membrane plenty, and we will some more. We won't go too much into it. We talked about steroids a lot too. What makes a steroid a steroid? Well, it's a lipid. Why is it a lipid? Okay, well, it's a lipid because it's all carbon and hydrogen, except for a couple little functional groups. Oh, let's see here. Here's cortisone. Here's, an oxygen here, an oxygen here. Where's estrogen? Oh, sorry, ladies. I didn't put estrogen on there. Oh, wait, no, I did. This one's estrogen right here. Here's testosterone. Isn't it crazy how, like, one little double bond here compared to there, and a hydroxyl group compared to, what is that one? This, what do you call it? An aldehyde? Is that what that is? A carbon that's bonded to two carbons and a double oxygen. I don't freaking remember. Let's call it an aldehyde. Okay. Kurt or Ethan or Rock, help me out. You guys are in AP Chem. But anyways, let me know. And then give me a yeah, Verdi. Okay, so these are all hormones. You've heard of cortisone. It's a measure of how we measure how stressed you are. Your cortisone levels. Okay. All of these, that's an email. Okay, all of these are based off of cholesterol, right? We ingest cholesterol and then we turn it into testosterone or estrogen or whatever else we need to. Okay. Oh, now we got to get to waxes. Sorry. There you go. There's those guys. Right? The different little tiny doses change how we think, behave, and grow muscle, fat, and bone, and hair. Okay, so wax is last thing. They're another lipid, and we're done. They protect things, right? They're waterproof. When I say protect, earwax, right? Keep my brain from getting wet, okay? By having lots and lots of ear cheese. Nasty. Okay, so plants are covered in a cuticle, right? If you notice the water beading on this, like maybe you'll go and wax your car today if you have one, or wax your parents' car, or your neighbor's car, right? And watch the water bead because it's going to rain. Weather report, right? Tuesday, Wednesday, Thursday, Friday, Saturday, Sunday, we're getting rain. All right, so go outside today because it's going to be rainy. I think for the next 6 days as we check the weather to see, can we get the children out of the house? Can we lock them out of the house? Nah, it's not quite warm enough. Okay, who knows? Annabelle, if you watch this in 10 years on my YouTube channel and I'm gone and you're learning AP Bio from YouTube, I didn't mean it. All right, so anyways, kids, let's stop right there, okay? Stop share. Let's Hi, everybody. I'm back to seeing you.